Hello, friends. I am Sandeep Kulkarni from ImageProVision Technology. I would like to thank the organizing committee for giving me the opportunity to talk on this prestigious 9th annual Global Pharma Regulatory Summit and giving me the opportunity to talk to this august gathering audiences who are sitting all across the world. You all are listening to the eminent speakers since morning, and you will be listening in coming sessions as well. I'm going to talk to you on a very specific topic, that is regulatory challenges on particle analysis process. So I'll just share my screen. I hope you are able to see the screen now. So, as I told, regulatory challenges in particle analysis process. Let us first understand why we are interested in particle properties. I need not tell you, you all are experts, but I've just noted down a few of the points for which particle analysis is pretty important, like better control on product quality, improve product performance. Now, if you have a total control on your product particle and properties, your end product will also be in the same specifications as far as regulatory requirements are concerned. So it's important that you take the particle properties very, very carefully. So there are other important aspects like troubleshoot manufacturing and supply issues, better understanding of products, ingredients, and processes, optimization of efficiency of manufacturing process, yield improvement, and most important, to comply regulatory requirement for which we all are here. Now, we all know what is the role particle characterization play in pharmaceutical industry. In the pharmaceutical industry, the particle size distribution, and particle shape, that is morphology of API, is known to strongly affect the stability and aesthetics of the formulation, which is very important when it is concerned with the regulatory requirements. So the size and shape of particles used in pharmaceutical drug product, not always, but most of the time, impact the dissolution rate of the drug. And all these are the important aspects when it comes to the regulatory compliance and regulatory challenges are concerned. So although there are various ways we do the particle analysis, and most popular way is doing it with laser diffraction technology, but here I'm just focusing on the latest innovative solution, which is automated microscopic analysis with the help of digital camera and software. So this is a microscopic way of particle analysis. I will talk on the system in detail in coming slides, but although it's an innovative solution, but this is what we have been able to have the customer list like this. So most of the major Indian customers and a lot of multinational companies have been using the system for both in terms of R&D for their new drug development, and in QA and QC for drug production or quality assurance. So coming to the main topic, US FDA queries in particle analysis process, first of all, for the A&D approval, that is in R&D space. So what I've done is that since we are working with so much of eminent customers, and we are engaged in various query resolution whenever US FDA or any other FDA is raising, so we are involved in answering this question. So what I have done is, in this particular particle analysis process, which kind of questions US FDA is asking. So I've just noted down all those questions, and let us try to see that how we can tackle these queries. First is like, please describe the difference in globule size and particle size distribution. Now this particular process not only gives the particle size, but it also gives the particle shape. And with the further upgraded versions, you can also differentiate between different particles like API, excipient, or API globules. So when you are giving the analysis for globules and particles, US FDA or any FDA would be interested how you are doing this differentiation of particles and then reporting the particle size distribution differently. So whether your system is capable of giving those answers, that is one of the important aspects. Please describe the calibration process of converting pixels to micrometers. Now, since we are in this process, we are working on the images of the particles. So converting those images, that is in terms of pixel, into micron. So whatever size we are reporting, so auditors would like to understand how we are converting those sizes, whether those sizes are accurate or not. So you need to understand the calibration processes. Please describe the impact of parameters chosen for image processing and discuss your procedure for excluding or accounting for touching globules and globules along the edge of the image. Now, it is as good for globule as good for particles as well. So the tendency of every particle is to come together and form the connecting particles or the agglomerations. So whether your system is capable to separate out the analysis of isolated particles and agglomerations, is your system capable of that? So that is one of the aspects you have to see, and whether your system is able to generate different statistics for agglomerations and isolated particles. Similarly, there are various other questions which generally the FDA people ask. One more question, the last one is, how are you comparing various parameters of your DP sample and RLD sample? Is your system able to generate statistics or comparative reports giving these two comparisons on individual parameters? So these are the questions which, there are multiple questions. Again, please improve the optical quality of your images so that API particles are in focus. So they would like to understand whether you have taken the correct images. Now, who will decide whether this image quality is correct or not? Is your system capable to identify that this image is good enough for the analysis? Because if your image is not correct, your analysis will not be correct. And in that image, whether your particles are visible correctly or not. So regulatory authorities are concerned, or they want to understand how you are doing this. I will not go into details, but again, C, please describe the impact of different sample sizes on D10, D50, D90 matrix. So if I'm taking 10,000 particles, my D10, D50, D90 is what? If I take 50,000, is it changing? So with the different sample size, my consistency is there or not? So US FDA is concerned about that. So let us understand how we can avoid some of these mistakes, which we normally do. First is understanding the calibration and validation process. I have seen that most of the people are worried about the date on which the calibration is to be done, the process to be followed, the report is to be done, signed properly. But I would also request to spend some time on understanding the process as well. Once you understand the calibration and validation process properly at the fundamental level, it will become easier to face the auditors. Prepare good sample for analysis. The sample is, if your sample is good, your reports are good, your analysis is good. But who will decide whether it's a good sample or not? Is your system is capable to differentiate between a good sample or a bad sample? Keep images for reference. Now, when you are doing a manual analysis, then of course it's very important. Whatever images you are taking, are those images good enough for analysis? So you keep a reference image that whenever I take the image, it should look like this. But if you have a system which on itself can decide whether the image is of good quality for processing, for analyzing, then your headache is slightly lower and you are able to convince the auditor more confidently. There are multiple other things like set parameters, such that you are capturing the correct images, identify classification parameters, as I told you, API and globule, or API and excipient classification. And the one is have confidence in your system is very, very important. Now, what I have seen is that the moment you get a result which is not as per your expectation, the first thing you want to conclude is that there is some problem in the system. Let me tell you that no system will behave abruptly unless we do some process differently, not as per the process, or we have not set the parameters correctly. So whenever you are getting some results which are not expected as per the expected lines, please go back and check your process if I have done all the steps correctly or not. If at all there is problem, you can always ask to the manufacturers for the help. Now, coming to the second part, which is US FDA queries during the production processes or quality processes. So our bad news, that is 483 and warning letters. So in the particle analysis process, again, I've noted down most frequently inspection observations which US FDA has put. Now, when we look at in details, it is valid for all, not only for particle analyzers, for all other processes. Most important is procedures are not in writing, fully followed. So one is that whether your process SOPs are in really good SOPs and are you following it correctly. So most of the time, this is where written processes are not elaborate. So you don't have to create SOPs or the processes which are very general kind of thing. So based on every process or every product, every equipment, you have to be slightly detailed SOPs there and all parameters are captured there. Then generally you are safe. All other like testing and release for distribution, computer control on master formal record keeping is all this. So most of the common inspection observations, you all would find that these are the simple steps, but there sometimes we are not able to follow the simple steps. Now, what we can do in this, as per our experience, understanding the 21 CFR Part 11 standards is important. So these are the guidelines, but based on your product, based on your equipment, you have to interpret and put the standard in proper manner. Be careful while installation. During installation, a good team or the person should get involved. What is the antivirus you are using? Restricted folders, data, images, reports. Now, IT is a good enabler. It has eased our life quite a bit. It has simplified our processes, but it also comes with some challenges like antivirus. We are already facing a biological virus, but IT is prone to various kinds of viruses in past as well. So you have to choose your antivirus system very correctly. Also, you should take care that your antivirus should not affect the software which you are using. So it should not interfere with the analyzing software. So detailed parameter set, SOPs, privilege setting, and all other important aspects. One more aspect I just want to highlight is keep your system upgraded with latest version. I know that going for the latest version of upgrade, you have to go through change notes and very complicated processes, a lot of paperwork, approvals, and that is why most of the system generally avoid for upgrades. But what is happening is IT, unfortunately, is such a dynamic environment that every day there are changes coming in. Sometimes there is changes in the operating system, sometimes changes in the systems, sometimes changes in the environment. So it is always, we advise that keep your system along with the latest version, because you don't want to change. You are happy with your current version, but unfortunately the environment around you is changing, and that's why you have to keep upgrading your systems. And again, have confidence in system, I've already told. So these are the challenges which is thrown by US FDA or any FDA on the particle analysis. So do we have any technique which can provide us particle size, shape quickly, accurately, and most importantly, fulfilling all regulatory requirements? So as I told you, I'm going to talk about the system. It is microscopic based particle analysis system, which has got a motorized stage XYZ. It has got a digital camera and a patented algorithm, which does the analysis part. So let us quickly understand what is the analysis of it. So in the left side, you will find the image which comes from the microscope, and what the software does. First of all, it identifies each and every particle. Then it classifies whether it's a isolated particle or an agglomeration. It is done through a patented algorithm. This solves one of the major query which auditors ask, whether this is a real particle size. So it generates the statistics for isolated particles and agglomerations separately. And now within isolated particles, it also classifies the particle as API, or excipient, or API or globule based on the size, shape, color, texture, intensity, and many other parameters. This is how the screen looks. It's a busy screen, but it gives all the information. Your live images are coming, statistics are populated on the fly, your graph is generated, histogram is generated, D10, D50, D90 is calculated on the fly. So you get all the information here. It gives a very detailed report as per the regulatory requirement, unique number, date, time, user information, and along with that, you get also the path of the analysis, where it is saved. But there are few more challenges which we can address, regulatory requirements which we can address with the newer technologies which is coming, that is particle size and shape analysis powered by machine learning and artificial intelligence. Now, it's a very complex system, but I will try to simplify it with a very simple diagram. Now, the first question was whether the image is good enough or not. So with this system, a machine learning engine works, and whenever you capture image, it automatically tells you that whether this image is correct for analysis or not. The second machine learning engine works on the particle classification or particle identification, whether this is API particle or excipient particle or globule, that it works on the machine learning. It automatically does. So the subjectivity is removed in this. All the data goes into statistical analytics, and it goes to the artificial intelligence engine. So as you are using the system more and more, it becomes more intelligent, and it solves a lot of regulatory challenges which you normally face in a manual system. So a lot of data is created, information of each and every particle is appearing there. So let us quickly look at how this system works.
There is no denying that accuracy and quality are key challenges pharmaceutical companies face daily. You want to increase productivity, but couldn't do due to the complexity of processes while maintaining the accuracy of data. So how can you fasten this process while maintaining the quality of product? Introducing ImageProVision's IPVP class software. IPVP class is microscopic image analysis system that includes microscope of any make or model, a unique HD camera, and a dedicated software for identification of particles. It is an easy-to-use software providing key features, such as particle size and shape analysis, identification and measurements of particle, morphological details of every particle, identification of agglomeration, auto-scanning of slides, particle classification, and 21 CFR Part 11 compliance. As the IPVP class uses a dedicated particle characterization software with particle size and shape analysis, it gives you more accurate analysis in just seconds, proving accurate and time efficient, reflecting in your increased productivity. And as it uses advanced microscopic particle size and shape analysis technique in place of traditional laser diffraction technique, it is also a cost-effective solution for saving your resources. So let's see how it actually works. Step 1. Take sample of any dosage form, like powder, liquid, cream, injection, etc. Prepare the sample on the glass slide and place it on the microscope. Step 2. Select appropriate magnification required for analysis. Step 3. Now, IPVP class will automatically take multiple images of different fields of view through XYZ motorized stage. IPVP class software works through its patented algorithm to generate a histogram, and D10, D50 and D90 graphs are plotted. Each particle gets identified and further classified as isolated particles and agglomeration, whereas isolated particles are further classified as API, excipient, etc. Statistics are calculated in terms of particle count, minimum, and maximum average size of particle. Separate statistics is generated for each type of particle. The output is a well-designed report that provides representative image, histogram, input parameters, particle statistics, distribution values, and morphological details, which contains image of particles and other morphological parameters. IPVP class software provides user identity, electronic signature, detail audit trails, non-editable reports, data encryption, and data retrieval, complying to all clauses of 21 CFR Part 11. Large numbers of customers across the globe have used and endorsed IPVP class as a complete system for particle size analysis and particle classification. For more information, visit www.ImageProVision.com.
So thank you very much for this session, and I hope it is useful, and I'm open for the question and answers session. Thank you.